So let's go ahead and start in with today's lecture. We have been talking about the clinical treatment of cancer. We've spent the whole semester unraveling and understanding the cellular pathways that cause cancer, and also appreciating the context of the in vivo tissue environment in which cancer cells transform, become cancer, and the various challenges along the way that the tumor faces, ultimately leading to metastatic cancer. And so that's why the last couple lectures, we've now been talking about a broader picture about how we treat cancer and the types of challenges and considerations that go along with that. We learned about the idea of surgical intervention, the effectiveness of radiation, and we are focusing now on this idea of drugs, right? Different types of small molecules that can be used to treat cancer. And so last time we stopped with this slide, and this was a slide that introduces this idea of, if you want to design a brand new drug for treating cancer, right, so a de novo drug, not something that's based upon a compound structure that's been known for decades. You know, we looked at a couple of examples of those. For example, the Taxol, that complicated organic compound with lots of rings and things that came originally from the bark of the Pacific yew tree that was discovered in the 70s as part of this big natural product screening initiative by the NIH. Right? And then we learned how in the 80s and 90s, big pharmaceutical companies learned how to artificially synthesize Taxol in the lab, right, and make it more available as a drug. And that drug interferes with microtubule regulation in cells that are undergoing mitosis. But we said, you know, what if you actually want to start and design something new from scratch, rather than trying to just screen hundreds of thousands or millions of little compounds, right? What if you want to take a rational approach? Meaning, what if you have a target already in mind and you want to come up with a small three-dimensional molecular shape that targets that inside a cancer cell? So that's what we mean by rational drug discovery. So last time we stopped with this slide, and we said that there are a number of different considerations, everything from, number one, feasibility of the target, meaning you have to be able to identify what it is you're trying to target. Is this a receptor? Is this an enzyme? And then even if you've identified, yes, that is a critical protein in a particular pathway, you have to ask yourself, well, what on this protein am I trying to target? Is it a ligand binding site? Is it a catalytic site of an enzyme, etc.? Then you have to be concerned with specificity. If you come up with a three-dimensional shaped little molecule that binds to this target, how do you know that it's specific just for your target protein and won't bind something else inside of the cell, which might be potentially far more dangerous than beneficial? Then bioavailability, right? We said it's one thing if you put this compound in some media on a dish of cells on your lab bench and you say, oh, the cancer cells in the dish die. But what happens when you actually put this drug inside of a living in vivo environment, whether it's a mouse or a human? Right? What happens to this drug? Does it become metabolized? Does it get broken down right away so it's actually useless because it doesn't reach the tumor? Right? So that has to do with different types of pharmacokinetics, pharmacodynamics. We'll take a look at those today. And then lastly is efficacy. Even if this drug does have a nice pharmacologic profile in the body and it's not super toxic, it doesn't have a lot of off-target effects, is it still going to work against the tumor in this real in vivo tumor environment compared to a plastic dish? So let's explore those today. And I think this is a nice way to kind of wrap up our semester, because we're going to apply many of the things we've talked about. One way of identifying a target that you might want to design a drug against is to have some good biological rationale, right, for why this particular protein is a good target to look at. So in the last decade or so, with the advancements in high throughput sequencing, as well as computational technologies, being able to crunch a lot of numbers on something like your laptop now that maybe 20 years ago would have taken several large desktop PCs all working together to do, right, enables us to do many types of functional genomics that we couldn't do before. And that has really aided in our ability to identify good targets for designing new drugs. So this data right here is looking at a range of breast cancer data using functional genomics as a criteria for trying to identify good protein targets. So the way you look at this here on the left, right, this is essentially like a heat map. You can see on the y-axis, it says 295 tumors, right? So that means you can pretend, this means you took 295 different breast cancer patients and you took samples of each of their tumors. So you have 295 different tumors from 295 different patients. Now, right off the bat, you can imagine with almost 300 patients, not all of their cancers are going to be or behave the same. Right? They're going to be very different, not only in the cancer biology of the tumor itself, but also in the clinical outcomes of this patient. Some of these patients are going to have better prognosis, better survival than others. So you want to wrap that all in here and take a look at, can you actually identify protein targets that stand out as being relevant or important to how these different categories might all be put together? So on the y-axis is 295 tumors. That means each row going across is one tumor. So you can see, if you look very closely, each row going across is made up of little rectangles, tiny little rectangles, right? If you count it across, you'd find that there are 70 rectangles that go across. What those represent is 70 different genes that are being evaluated in each of these tumors. Those 70 genes represent 70, let's say, proteins that you believe might be relevant, might somehow influence whether this particular tumor ends up being aggressive, not aggressive, whether this influences the patient's better survival or poorer survival. So for each of these tumors, you go ahead and you look at expression levels of each of these 70 genes. So that means, let's start from the top again, right? As you're walking across, each square would be the relative expression level of that particular gene out of the 70 genes that are being looked at. Here on the bottom is your legend or your scale. So for genes, you compare the tumor with normal tissue that might be nearby the tumor inside the same patient. Right? So that means for each patient, you have some normal tissue and you have the material that came from the tumor. And you say, for each of the 70 genes in the tumor, how does the expression level of that compare with that same gene in the normal tissue? Look at the legend down here. If there's no real change in the expression level of that gene in the tumor, then you maybe just give it a black color, right? If the expression level of the gene is actually lower in the tumor compared to the normal tissue, you assign it a color that's more and more green. On the other hand, if the expression level in the tumor is higher than in the normal tissue, you give it a color more and more red. All right. So what you imagine then, okay, so that's where the high throughput sequencing comes in, right? Now it's very feasible that we can actually do this type of sequencing on 300 tumors. But there's more to it than that, because you can imagine, let's say around tumor 150 right here. And let's imagine that we're just looking at this row across. This is what you might imagine you would see, right? As you go across, some of these dots are red over here, some of them are black, some of them are green, then this one's kind of red again, and then blah, 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 right? It would be kind of all over the place because these 70 genes are just kind of randomly lined up here. This is where the computer comes in then. Because if you just made a big block of data like this without the rows being organized in any sensible way, the whole thing would just look like a big smear, right? Because it'd just be random dots of red and green and black everywhere. But what you can tell the computer to do is organize now this in a visual way that actually reveals patterns. So in this case, let's say the computer starts off with this sample, the top row here of 70, right? And the computer says, okay, in this particular one, the first few genes were very bright green, right? Then what the computer does is it looks through the other 294 potential rows and says, which other rows also start off bright green? And it starts lining them up based on that. So the ones that start off bright green are put together, right? And as you go down, you'd see fewer and fewer of these tumors have bright green to start with. And eventually it gets to the opposite. As the computer is organizing it, you end up with the tumors that start off bright red. And so the computer can do this. I mean, I guess you could do this with the naked eye, but obviously that'd take a long, long time. But the computer can just do it like in a few seconds, right? And what you see is when you arrange the 295 tumors like this, patterns begin to emerge, right? You see a cluster of these tumors here. Let's say this roughly, what is this, like 75 or so tumors up here that started off bright green over here, end up having bright red on this side, and vice versa on the bottom, right? Then what you do is you say, of these 295 patients, right, because each one of these tumors came from a patient, as you follow them over time, so this is a long study, you can determine which patients failed treatment earlier, which ones had a poorer prognosis, which ones had a better prognosis, meaning which ones had tumors that became aggressive and metastatic really quickly versus which ones of these people had tumors that stayed relatively good for a longer time, right? And you can actually figure out, you know, oh, okay, this dotted line would be the cutoff. It turns out that the patients represented below that line had a poorer prognosis. The patients above that dotted line had a better prognosis, as defined by something like the metastases. So in this case, poor meant, you know, this group had more metastatic lesions over time. This group above the dotted line had fewer metastatic lesions, right? So hopefully if you see this, you can see how all this data comes together, and it allows you to be able to get a sense then of what these 70 genes represent, right? And so you might say that certain genes that are underexpressed in the tumor, right, are actually a sign of a good kind of prognostic indicator. Whereas these genes over here on the right, these genes, when they're overexpressed, are also associated with a better outcome of this tumor. And then the opposite would be true, right? The genes over here, down in the lower left, that tend to be overexpressed, where the ones on the right are underexpressed, that gives you a poorer kind of prognosis, right? So that might mean then the genes that you're interested in looking at might be the ones that are, for example, in the very corner down here and the very corner over here, right? So out of 70 genes, you might not care so much about the ones that happen to be over here. You care about, oh, what are these genes that happen to be the ones that are in this corner and this one, because maybe those are good targets to look at in different pathways. And this data over here, this is a Kaplan-Meier plot using this data and showing you how they were able to follow the good signature patients versus the poor signature patients, right? And how they were able to then say that this gives you some idea about good protein targets to try to understand better. Right. So this can be very useful for types of cancers where the histology or the appearance of the cancers are very ambiguous. We've learned this semester that one of the problems with looking at advanced tumors is by then they become so messed up that oftentimes you cannot tell anymore, right? What you're even looking at. What was the original tissue that gave rise to this mess? And of course, one thing that I've been trying to encourage you to think about is if you can understand the original tissue that the tumor came from, then you have a better idea of certain pathways in those cells that might make for good targets for some sort of therapeutic intervention. So here's an example of an ambiguous cancer. So B-cell lymphomas, so these are diffuse large B-cell lymphomas or DLBCLs. There are 3 types of these large B-cell lymphomas that patients present with in the clinic. These are known as primary mediastinal, germinal center, or activated B-cell-like. Unfortunately, when you look at these 3 types under the microscope, the pathologist who's looking at the biopsy would pretty much see the same thing. It just looks kind of like this. Right? And so you have a very poor ability to really figure out when the patient's presenting with the cancer in the clinic, which of these subtypes you are dealing with. This is important because these 3 subtypes have very different clinical profiles in terms of how patients progress. You can see here, right? This is a Kaplan-Meier looking at overall survival over 10 years, starting from, let's say, the time that the patient first presented in the clinic. You can see that of the 3 different subtypes of large B-cell lymphomas, right, this PMBL, or the primary mediastinal subtype, patients have a better 5-year survival rate compared to the worst one is this activated B-cell-like subtype, right, the blue line here. So you can see the challenge, right? Because when the patient first presented in the clinic and they took a biopsy, they sent it to the pathology lab, the trained oncologist, pathologist looks at it under the scope. She cannot tell, right, which of these 3 subtypes there are, but yet it's important to know because that's going to have a big influence on the future of this patient. So we can use these types of functional genomics experiments, just like in the previous slide when we looked at the breast cancers, to look at which genes out of a panel of 70 genes are overexpressed and underexpressed. You can do that same thing, do the same type of study, but look at a panel of genes that you believe are associated with B-cell lymphomas in general, and then group the data by the 3 different subtypes. So all 3 subtypes, the PMBL, the GCB, the ABC, they have a common microscopic appearance. But when you do that type of heat map plotting of these genes, right, you can see really clear grouping begin to appear. So for example, in the PMBL subtype, these genes over here are the ones that the computer organized, right? After the computer lined up all of the data, these are the ones that are highly expressed. And then, so that would be this set of genes here, right? That are highly expressed in PMBL and not so much in, for example, ABC. Then you have, as you move down, you have this group of genes, right, that's highly expressed in the GCB subtype of B-cell lymphoma. So that would be these genes here. And then all the way down, you'd have this group of genes that are the ones that are highly expressed in ABC subtype of B-cell lymphoma. So now, even though visually under the microscope, it's kind of hopeless to tell these apart, if you know certain genes can serve as markers for different subtypes of this cancer, now you can actually use this, right? Because you can take the biopsy material and actually probe for the expression level of particular key genes based upon this chart here that will reveal to you, is it most likely that this patient has PMBL, GCB, or ABC, right? And not only that, but of course, this type of data is very valuable because now, if you want to focus, let's say, drug design on targeting the most sort of lethal form, the ABC subtype, now you know, right? It's this set of genes here that are the ones that are highly overexpressed. Maybe those should be the subgroup of targets that your research group focuses on trying to understand how these pathways work and maybe how you can intervene, right, or target these. So for example, right, so here's, based upon this nice colorful red-green data, you can put together something like this, right, where you can take some of these red ones, right, and actually figure out, okay, those genes that are shown here, right, which ones, what pathways they are actually part of. So for example, in GCB, right, this subtype, one of the genes that was identified very commonly has to do with BCL2 translocations. Or down here, you can see that in GCB subtype, you don't have constitutive activation of NF-kappa B related genes, whereas in ABC, you do. So I think my computer, unfortunately, just crashed here. Let me see if I can. I don't think I have to stop the meeting, but I just close my PowerPoint and start my PowerPoint up again. So hang on just a second. I'm going to start up my PowerPoint again, and hopefully it'll behave this time. Okay, so I'm going to go back to share screen. Back to that. Okay, good. So hopefully we're back on track here. Good. Okay, sorry about that. So let's look at this finding down here, that in GCB, you don't have constitutive NF-kappa B activation of that pathway, but in ABC, you do. Right? So not surprisingly then, if you go ahead and you look at a drug that inhibits NF-kappa B pathway, so in this case, there's an inhibitor that's a drug that's a I-kappa B kinase inhibitor, right? So this particular drug interferes with the normal inhibitory signals, the signals that normally block the I-kappa B from turning off the NF-kappa B, and so then it relieves some of the ability or to prevent the I-kappa B from getting to the NF-kappa B, and so as a result, the cell itself is able to try to turn off the NF-kappa B more effectively, right, by using this drug. And you can see, sure enough, if you look at the viability of cancer cells, this is, I think, in vitro data, if you treat with this I-kappa B inhibitor, this I-kappa B kinase inhibitor, you can see it's the ABC subtype of these B-cell lymphomas that are the most sensitive to this drug, right? And that makes sense because it's in the ABC type where the NF-kappa B pathway is being improperly amplified, right? So you could say, well, you could have by chance just stumbled upon this, right? You try a whole bunch of different drugs and you find this data, but that's not very efficient, right? In this case, you were able to use this type of data to right away identify a difference between the GCB and the ABC subtype, and therefore that led you to say, are there drugs available that would be relevant to this particular difference? And yes, there was, and you tried it in the lab, and it proved your hypothesis, right? That yes, this drug could be more effective in treating the ABC type of lymphomas compared to the GCB type, right? All right, so when you get excited about this idea of, oh, good, I've identified a protein that seems to be very relevant for the cancer that I'm interested in, right? Now I want to go about designing a drug against it, I don't want to use necessarily a drug that's already available. I want to use something that I come up with, right, in my research lab. So now this is the next problem. So let's say, for example, we're looking at receptor tyrosine kinases. So I don't think any of you would be surprised that receptor tyrosine kinases are a very, very popular target for anti-tumor drugs. So here we have an anti-ERB2, no, sorry, this green is the HER2 receptor. And below it are many of the proteins that we've studied this semester that have to do with how the activation of the receptor gets transduced into cellular signaling pathways that ultimately end up in the nucleus, turning on things like proliferation, survival, cell cycle progression. So hopefully there's some very familiar proteins here, right? So here's our SARC, here's our RAS, here's our PI3 kinase, AKTs, the MAPK signaling, etc. So any of these proteins throughout this pathway could be a potential target, right? You can design a drug that's a small three-dimensional molecule that somehow interferes with the ability of a protein, one of these, to properly signal the next protein in the chain. And all of these labels in pink are real examples of different classes of drugs that have been designed to target these different proteins in the pathway. I think this is really neat when you look at this, right? And it makes you realize this can only happen if you know the different components of this pathway, right? And let's say from your functional genomics sequencing data, those red and green heat map things, you've identified some new genes that maybe previously weren't considered part of a pathway that's relevant to the cancer that you want to study. And so you get excited because maybe that's an opportunity for you to design one of these drugs like you see here. So the problem then becomes specificity. So you've located a nice target, right? Now you have to actually figure out what the target space is that you're trying to target on the protein, right? And is there an issue of specificity? Specificity is a big problem, right? Because you might say, well, the target space on, let's say, an enzyme that I want to have a small molecule bind to and kind of mess with is maybe the catalytic site, right? That makes sense because if something jams into the catalytic site of the enzyme might not be able to function properly. And that might be a great way to disrupt a signaling pathway. But the problem is catalytic sites of enzymes are highly evolutionarily conserved. One of the analogies that I like to use is in my Bio 275 class, I always say enzymes are like a stapler. There are many variations of staplers out there, small ones, big ones. This is my swing line stapler, a nice heavy one that I use on my desk, right? One thing that the majority of them have in common is the catalytic site, right? This is where the actual stapling gets done, where you load the staples inside there. And they are pretty much all the same shape. Why? Because they're designed to use the same sort of generic staples, right? So you can go to Home Depot or whatever and buy the same universal kinds of staples, right? And a whole bunch of different staplers use this type of staple. That's because once the particular shape of the actual stapling part, the stapling mechanism was invented, that pretty much stayed the same. You can mess with the other parts of the stapler, right? Make it bigger, stronger, or smaller, more portable, but you don't mess with the catalytic site. So that's true with the evolution of enzymes as well. Here on the left is a panel of 9 different kinase enzymes. Right? And in blue, these are the more common serine-threonine kinases, right? Remember 99% or whatever of the phosphorylation events that happen inside of your cell involve phosphate groups being moved by one of these kinases onto a serine or threonine target in a substrate protein. Right. Here in orange are the tyrosine kinases. So these are the ones in our cancer class we learned are kind of special right? Because these are the ones that are like the VIP ones. They do tyrosine phosphorylation, right? Which is highly conserved and specialized for these important pathways that control cell proliferation, survival, cell cycle progression. But if you look at these 9 structures, they look the same, right? That's because these are 9 different staplers, pretty much, where the catalytic site, oh, so I should mention, right? These are not the complete proteins. These are just the catalytic sites of these 9 different kinases, right? So evolutionarily, they look pretty much the same. So if you look at a structure here, let's say that you take the 3-dimensional space filling model and you overlap a couple of these, you pick a couple of these from the left side of the screen here, and you overlap the space filling models, and you say where the shape is similar, we will make it gray. Where the shapes deviate a little bit between the two, you make it green, right? And this really shows you evolutionarily how conserved these kinase domains are. So the problem is you go ahead and you design, let's say, a small compound. Here is an example. This is an ATP analog, meaning this is a molecule that kind of is the same sort of shape as ATP, artificially designed in the lab. But because the shape of it is slightly different than ATP, it can fit into the ATP binding site of this enzyme, right, where ATP would normally go, and then the enzyme would normally break the ATP to use that energy to then act on some other substrate, right, to take the phosphate group from the ATP maybe and put it somewhere else. But because this synthetic drug actually is not ATP, it fits in there well, but the enzyme won't work, right? The problem is you go ahead and you target one of these. Let's say you're trying to target SARC, right? You design this and you're like, wow, this is great. It fits in SARC, and then SARC doesn't work anymore, right? And you think like, wow, this is great. And you see dollar signs and you're all excited. But the problem is what? This same drug is going to also inhibit the rest of these kinases as well, right? Because it fits in that same pocket. So this is why specificity becomes a real big issue. If you can design a particular shaped small compound that is very specific in fitting into a target space, now you're talking. Right? So this drug, Gleevec, or imatinib, targets the BCR-ABL oncogene that we looked at earlier in the semester. Okay. And so Gleevec, you can see, this is the Gleevec compound. Well, this is it right here, right? This interesting kind of S-shaped multi-ring compound. It fits right into this pocket inside the BCR-ABL oncogene, and it prevents the oncogene from activating. Okay? So maybe if you've discovered a small structure like this that fits in, you can improve the effect of that structure by messing around with putting different R groups at different locations. Right? So you want to do that so that you have to be careful. If you change the different R groups that are hanging off of this compound, you want to make sure that that doesn't actually make it so the compound no longer fits in the pocket that you want it to. So you want it to still be able to fit in the pocket, but maybe by having these extra R groups hanging off, you help this drug prevent the protein from then activating. So you can see here, circled in blue, the Gleevec has an oxygen right here, and in orange, there's this nitrogen hanging onto a methyl group, right? And you can see where those are in here. So this is the orange group, this is the blue group. And what this does is it helps cause very tight binding of the Gleevec compound to the BCR-ABL oncogene, right, which is represented in green. This is the ribbon diagram of the surrounding spaces. So with that kind of specificity that you improve upon in the lab, right, maybe you work with like a medicinal chemist, organic chemistry person, and you say, can we try adding these functional groups here? And then they go to their OChem lab and they figure out how to actually add those R groups, right? This is how you get a very specific, highly effective drug. So here, for example, we're looking at different types of kinases that might be potentially targeted by a kinase inhibitor like this Gleevec. So here in blue is the ABL, right? That's part of the BCR-ABL oncogene that you're actually trying to target. That's your goal. But these are some other closely related enzymes. So even SARC, right, is kind of sort of similar to how the BCR-ABL looks. But you can see, because you were able to finely tweak the different R groups that are attached to this drug, you're able to really increase the specificity of this drug to bind just to the ABL oncogene. So in blue, so this data, what we're looking at is as you increase the amount of drug, this is in vitro data, so this is a dish of cells in the lab, or several different dishes of cells, and you treat each one with this Gleevec, right? And then you go ahead and you analyze the data, and you actually see how the activity of these closely related enzymes are affected inside the cell. And so this y-axis is a little confusing because you can see it's actually the percent inhibition of kinase activity, meaning the higher you get to 100, it means the better you're getting at fully inhibiting this particular kinase target. Right? So you can see with the Gleevec, very clearly, the blue line, you're doing a wonderful job of inhibiting specifically the BCR-ABL oncogene, whereas the other closely related, similar three-dimensionally shaped kinase enzymes are less affected by it, right? So for example, SARC, right? So this is great. This is how you try to improve specificity by tweaking your compound. Okay? So how do you come up with these compounds? Well, like I said, you first do in vitro testing. So you might have, for example, a dish of healthy cells. So these are some mouse cells that are on their way to becoming lymphocytes, B cells, right? So these might be thought of as being the precursor of a B cell lymphoma cells. This is when they're still normal and healthy. You might want to first test, does your drug, like Gleevec, actually, is it toxic to the cells, right? Because if it's something that's toxic to the cells, then maybe you're not going to get anywhere with this drug if it just kills cells. So here you have these pre-B cell lymphocytes. What you do is you try to start stimulating them to differentiate into B cells. So you add IL-3, interleukin-3, and this is sort of your control, right? The cells thrive, they proliferate, right? And they grow. So that's what they normally healthy would do. You remove the IL-3 entirely from the media as another control. And sure enough, the cells die through apoptosis because they need this normal IL-3 signaling inside the cell for their differentiation. So then what you do is you say, okay, so now I know that these cells behave like normal. We can stimulate them to differentiate with IL-3. We can kill them if we take away this IL-3. But now what if we differentiate them using the IL-3, but also have our Gleevec drug in there? Is there going to be any impact? Right? And you can see the answer is no. So this is nice because this means that the Gleevec itself is not somehow toxic to the cells, and it does not seem to interfere with the normal healthy cell's development. So this establishes then a baseline response model, right, and rules out off-target toxicity. Then what you say is, now let's turn this into a cancer model. So with these IL-3 cells that have differentiated into B cells, you go ahead and you put in the BCR-ABL oncogene, right? So now the nuclei are like red because now they have this oncogene in them, right? And now you go ahead and you say, now let's take away that IL-3 signal, which is normally crucial for them to stay alive, right? You take away that IL-3 signal, ooh, the cells don't die anymore, right? They don't die because they're transformed. They've been transformed by this strong oncogene that you put in, the BCR-ABL. So then you say, oh, okay, good. Now we have our model. So this is our model of B-cell lymphoma, right? To that, you go ahead and you add the Gleevec. Right? You add the Gleevec and you see, oh, the drugs proliferate, right? That way you know the cells that are receiving the Gleevec that it somehow doesn't interfere with the normal IL-3 signaling, right? But the real kicker is, what if the cells don't have the IL-3, right? The ones that keep growing, the ones that represent cancer, you add Gleevec to those, and what you find is now the cells die in response to the Gleevec. Right? So this wonderful flow chart, I know it sounds kind of complicated, but I encourage you to go back, review this, talk your way through it. This is a really important cartoon because this really is the basis of how you do de novo drug design and discovery in the lab, right? And even if you're testing different drugs that maybe you have a drug that you know is used for some other disease, but you have a hunch or maybe some evidence from doing functional genomics types of heat mapping studies that this drug might actually be relevant to a particular type of cancer that nobody has ever thought of using that drug for, right? You would still start off using this type of experiment to test it out. Okay, so a number of cell lines that you can get from the National Cancer Institute are available if you're running a research lab and you want to have different cell lines representing different cancers for you to test a drug on. So we call this the NCI 60 cell line library. These are the most common cell lines that are used for in vitro experiments. So one drawback of this, though, is that these cell lines represent some of the most aggressive cancer cells that were originally isolated from an in vivo tissue source and then turned into a cell line that could basically last forever, right? Sort of like the story of HeLa cells, right? This idea of HeLa cells originally came from cells taken from a biopsy of cervical cancer back in the 1950s, right? But over the decades that they've been used, right, so only certain cells from that biopsy actually really aggressively grew in culture, right? And those have become the HeLa cells that we have now. So there's a bias. That means these cell lines that represent different cancers, right, not only represent different cancers, but represent very, very aggressive forms of those cancers. This data down here kind of illustrates this problem a little bit better. Let's say you take a group of patients, some who have more aggressive tumors, some who have less aggressive tumors, and you go ahead and you take biopsy material from these tumors and you try to introduce these cells into tissue culture and see which one of these cells actually keep growing and can be used as a cell line in the future. Meaning you can keep growing, passaging these cells, you can share them with other labs, you can freeze them, thaw them out, and use them. What you see is that the cells that end up successfully establishing cell lines in the lab came from the more aggressive tumors, right? Meaning the patients who these cells came from, those were the patients in this Kaplan-Meier plot who did much more poor in terms of their survival, right? The cells that came from the less aggressive tumors, right, you can see here, right, fewer of them, right, came from, or no cell lines were able to be generated by those less aggressive tumors. And those came from patients who did better. So this means you're biasing the creation of cell lines for cells that are super messed up and super aggressive. All right. So using these cell lines in the lab then might bias your results if you're doing experiments in little dishes. Why? Right? Because you need to be able to go ahead and move into animal models. It's one thing for a dish of cells to behave a certain way, but what if you actually try using your drug in living in vivo tissues? So mice, of course, are used for this. So there are several different genetically inbred strains of mice that are used. They are things like this Balbi C mice, C57 black 6 mice. These mice have different genetic backgrounds, right? But at least you know if you order BAL-BC mice, you know that these are genetically identical to every single other BAL-BC mice or mouse, right, used by other labs, or a black 6 mouse, right, that has a genetic profile. There are also immunocompromised mice, like the nude mice. Then you have this karaoke mouse here, but this is not really a cell model, right? Using karaoke mouse, that's a very unethical model to use. But when you use these mice, right, you can then try looking at how your drug data looks inside one of these. So one way you can do this is you can actually try to take living tissue from, let's say, a human body, right? Let's say that there's lung cancer here, and you take it out and you look at it under the scope. Right? Well, these two here, let's say this is an example of some prostate tumors that were taken for biopsy. Right? So this is a surgical specimen. So this is how the tumor looks inside of the human being. Here is similarly a surgical specimen taken from colon cancer. If you go ahead and you put them in mice, and you inject them in mice, and you try to grow tumors from them, you can see already the tumors that form in the mouse, they're similar, they have similar features, but they kind of begin to differ. Right? But at least you can tell they have the same sorts of morphological features as the tumor did in the human being. Right? So you say, okay, great, this is what I will do. I will generate the tumors in mice and then treat these mice with my drugs, right? But then you say, well, not so fast, because wait a minute. If the only way I can generate these tumors in the mouse is to go back to a human being and take tissue from it, that's not feasible, right? Because what? Maybe you get some tumor material from a human patient and you generate what? Maybe 8 mice, and you do your experiment, and then you have to repeat your experiment. What do? You can't go back to the patient and say, hey, I need some more of your prostate cancer, right? So that's why you need to actually use cell lines. So what you need to do is actually do an additional step, meaning you take the cells from the human, right, and somebody, maybe not you, introduce them as cell lines. You turn them into a cell line that can be perpetuated. And then what you do is at any time you need to generate more mice for your experiment, you thaw out aliquots of your frozen cell line cells, inject them into the mouse, and get the tumor to grow, right? So obviously this is advantageous because you don't have to go back to the original person, right? But the problem with that is if you inject cell lines into the mouse, even though they originally came from prostate or colon, by the time those cell lines grow in the mouse, they look like this. Right? And again, that's because you've biased yourself to cells that only successfully were aggressive enough or messed up enough to survive as a cell line, right? And so when you try to regenerate tumors from those, it may be that the tumors you actually end up creating in these mice for you to test your drug on turn out to be very far away physiologically and functionally from the original tumor type you're trying to target in a human patient. Right? So that's a big concern. All right, so just 2 more slides. I know we're kind of running out of time here. I'm sorry, I'm going over. So let's say you get to the point, though, where you've designed this drug. It works not only in dishes of cells in the lab, but you've now successfully done mouse experiments, and you're happy to say that the tumors that grow in the mice, when you inject your new drug, right, the tumors go away, right? So you're getting more and more excited. Now, maybe if you're lucky, you can start moving into human test subjects, right? So this is what we call clinical trials, right? So this is a very big deal because out of every new little compound that some research group comes up with and designs, maybe only 1 out of 100 such compounds ever make it to a clinical trial, right? And of those, the majority of them don't pass the first clinical trials. So clinical trials are designed as 3 phases: phase 1, phase 2, phase 3. Phase 1, the whole purpose of this clinical trial is not even to see whether or not it works on cancer in a person. A phase 1 clinical trial is just to determine, is this drug lethal, right? Will you kill people by injecting this drug into them, right? So phase 1 trials determine therapeutic windows. You can see here, you have 2 lines. The blue line represents the dosage of the drug that you need to get up to in order for this dosage to actually have any kind of effect at all inside of the human body, right? At the same time, you have this red line, and this red line measures toxicity. In other words, as you dose up the patient, you reach a certain threshold here, this black arrow, above which you begin to have really bad toxic effects, right? So that means somewhere between the dose that's needed in order to have any beneficial effects and the dose that you've reached to have toxic effects, that's your therapeutic window, right? So phase 1 clinical trial is meant to determine, does a reasonable therapeutic window exist? Phase 2 clinical trials then determine what type of targets are actually being targeted by your drug in the person's body, right? So in other words, you might start having different cancer patients involved in this trial, giving them different, so then you start giving these trial subjects doses of your drug that you determined is the therapeutic dose based on phase 1. And then you look at, are there any effects on various tumor types? Right? And then lastly is phase 3. Phase 3 would be, you've succeeded in showing that the drug is not super toxic, that there's a reasonable therapeutic dosage that you can give to patients, that patients' bodies tolerate it well, and you can see reasonable responses on target tissues. Phase 3 then, you really establish then, what are the particular on-label, meaning what this drug is meant to treat, right? That you can actually use to identify who are the patients out there who will benefit the most from this drug. So phase 3 establishes the significance of the drug treatment. And this is when you have very large and expensive studies involving many different institutions, hospitals, thousands of patients, right? And you try to see, does the drug work? So here, just to wrap up today, is an example of a drug called gemcitabine, or GEM, that was in clinical trials, phase 3 clinical trials, as a treatment for pancreatic cancer. Previously, the commonly used drug, it was this 5-FU or 5-fluorouracil, one of these analog drugs that kind of looks like a nucleotide, so that the pancreatic cancer cells, as they're rapidly dividing, incorporate this into the DNA and it messes them up, right? So now you compare, if the 5-FU is the gold standard of what you're trying to compare your brand new drug, the gemcitabine, against, you do a big clinical trial. One cohort of patients gets the 5-FU treatment, one cohort of patients gets the GEM treatment. And then you follow them over many, many months of treatment, and you see, is there any improvement? And as you can see here in this Kaplan-Meier plot, the test subjects who are on the GEM, right, their life expectancy was increased. Is it astounding increase? No, it's marginal, right? It increases their life expectancy maybe from, let's say, in the most extreme cases, from let's say 12 months to 18 months, right? But the amazing thing is that these days, if your new drug can extend the mean survival of a group of cancer patients by just a few months, 6 months, right? The FDA, the Food and Drug Administration, are inclined to approve your drug. Why? Because 6 months, at least it's something, right? It gives that patient another half year of life, right? To be with their loved ones, to get their things in order, okay? But I want to end with this because this shows you, this is about as good as we can do right now, right? And that isn't very impressive. A brand new drug that goes through all of that work that we talked about today, right, just to increase life expectancy by a few months. And that's why you guys, some of you will be oncologists, and your focus will be on designing new drugs. Right? So hopefully this will inspire you to really want to utilize new technologies, new strategies to make this a much more impressive story.